Thanks. My name is Allison Demas, and I'm a postdoc in the Bhatia and Alter labs. And I'm actually a parasitologist by training, and I'm going to share a little bit of my work with you guys and tell you how I came to be here in the Koch Institute and a Cancer Research Institute working with a bunch of bioengineers. All right. So our bodies are these complicated landscapes of many different types of cells that interact in different ways on different scales. And as scientists, our job is to learn how cells make choices, both good and bad, about what to do in our bodies. But asking these types of questions is actually quite challenging. It would be a bit like looking down at a video of all of the fans streaming out of Fenway Park after a game and trying to zoom in on one particular person and ask why they went one way instead of another. What motivated that choice? And can we understand how to motivate them to make a different choice? It turns out that asking these questions is actually quite hard. Where would you start? How would you do that? And in the Bhatia lab, we're known for harnessing engineering tools. Oops. Oh, sorry. It's not
doing what it's supposed to do?
No, sorry.
No.
Sorry.
No.
There we go. Okay. In the Bhatia lab, we're known for harnessing engineering tools to create deconstructed models of microenvironments in order to ask exactly these types of questions. It's one of the exciting aspects of being here at the Koch, where we have all of the materials and different people we need to use our bioengineering know-how to build systems to solve biological problems. So in our case, we use these tools to learn how cells make and repair complicated tissues like the liver, shown here from the organ all the way on the left, deconstructed down to individual cells on the right. We can ask questions about how different kinds of cells become tumors and how other cells, like those of our immune system, work together to fight them. So the biological question that I'm interested in is how our body is able to fight off an infection by an invading pathogen, the malaria parasite, that's transmitted by a mosquito and then invades and grows and develops inside the liver before bursting out, invading our red blood cells and making us sick. We want to learn more about this process so we can design new therapies, such as antibodies or vaccines, to help fight that infection more efficiently. And the way I've tackled this problem is to use one of the tools that we've developed that can grow human liver cells in a dish. It turns out that these cells only grow if you combine them in just the right arrangement and pattern shown here with other cells that are normally found in the liver. Our team has learned how to get malaria parasites to invade these experimental liver tissues, and now my job is to add in even more cell types, those of the immune system, so we can understand how immune cells and antibodies work together to fight malaria infection. Because if we understand how this process works, we can learn how to make it even better. And taking images, as we've seen tonight, is one way we can understand how cells work in systems like the liver tissues that I just described. And electron microscopy is a really powerful imaging tool that we can use to look at individual cells in exquisite detail. So I'm going to show you some of the images that we've taken here at the Koch Institute in collaboration with David Menkes and the NanoCore. This one right here is an example of a malaria parasite. And just to give you a sense of scale, while it looks rather big up there on the screen, the width of this parasite is actually almost 100 times smaller than the width of a single human hair. So this guy, even though it's that small, is actually quite deadly. And here's one of the immune cells that I've been working with. This is a natural killer cell, or an NK cell. It's one of our immune system's first line of defense, and they're highly cytotoxic, which means that they, too, are excellent killers. They're also highly abundant in the liver. And what you're seeing here is a series of snapshots of these NK cells as they become activated by different signals, such as malaria parasites or antibodies. And what you can see is that the surface of the cell changes. You get these protrusions that emerge, and it becomes quite bumpy. This is the image that you may have seen outside of one of these activated NK cells in particular. And these cells can be found in all of us. They're working around the clock to protect us from different invaders, pathogens like malaria parasites or malignancies like tumors. And so I want to end with these images here that I'm particularly excited about that show these interactions between NK cells and parasites. The NK cells are also quite excited to see the malaria parasites, while the parasites look decidedly less excited by these interactions. So we're excited to use the tools that are here at our disposal at the Koch Institute to better understand these interactions, what's going on with the NK cells and the parasites and why, and hopefully harness the ability of these cells to protect us from malaria and other diseases. So with that, I have a lot of people to thank, Sangeeta Bhatia and Galit Alter for their mentorship, as well as the members of both labs, with a special thanks to the malaria teams. Also a big thank you to the nanotechnology core facility here at the Koch Institute. I mentioned David Mankis, who helped me take all these images, but also to Abigail and Peggy for their help and our additional collaborators and our funding sources. Thanks.